Okay, so this is a project that is a collaboration between my group at South Dakota State University and Sangwon Jung, who is at Cornell University, and Leonardo Chamorro, who is at University of Illinois at Urbana-Champaign. We got this grant last year. It is on designing a novel mask, and initially it was intended as a response to COVID-19, but as we are going through the research, we believe there could be other applications as well. So the basic idea is this. So the masks that we had available when this big pandemic happened, surgical masks or N95s, they are designed not to resist this specific type of transmissions. Of course, they can resist them, but they are also trying to trap particles which probably don't carry that much of a load. Because, for example, if you think of N95, they can also trap the 0.2, 0.3 micron particles. So we thought maybe we can look at the animal world and see if we have better designs that we can capture in the masks. The reason being, we know that some animals, they have a very strong sense of smell. They have a very complex nasal cavity, and the aerodynamics helps to screen particles from the air, which the animal is breathing in. So we thought that maybe we can utilize that design. Our hope was, as a result, we can capture the particles that are the bad particles carrying all the pathogens, and yet the pressure drop that is needed to drive the flow through this mask won't be that high, which in other words means that these new masks would be more breathable than the N95s or the surgical masks. So the summary is we are trying to design a mask that uses this bioinspired design, should be more user-friendly, more breathable, and yet trap the particles that we need to be worried about. So on this slide, you can see a CT scan of a nasal passage of a dog. We try to look at the complexity of the airway, and the second picture on the bottom right, that's a visual of a single filter pathway, and around 300 of them would go into that circular filter that's shown here. We looked at a number of animal species, and we tried to figure out what is the tortuosity of their nasal passage. Tortuosity is the curved length over the linear distance between 2 points. And that's how we designed our masks. Our filters, it covered a wide range of tortuosities, which are similar to the ones seen in these high olfactory animals. So as I said, this is a collaboration between 3 universities. My group runs the simulations, the computational simulations. Sunny's group at Cornell, they are doing the biological characterization, looking at taking the scans from animals, figuring out their geometric parameters. And Leo's group at UIUC, they are running the physical experiments. So on the left over here, you have a schematic of the physical experiment. It's a pipe, sort of like a wind tunnel. We place the filter, the 3D printed filter in there, and then we drive air through the filter. The flow rate of air is similar to our breathing rate. We also embed particles in the upwind side, and we try to see how efficient the filter might be to trap the particles. And on the right over here, we have replicated a similar setup for the in silico simulations. This graph is a little busy, but what we see over here is, if you look at the top 2 lines, therefore N95s and surgical masks, the crowded lines at the bottom, therefore the new designs that we are developing for different tortuosity values. This implies that let's imagine that we are looking at the 60 liters per minute flow rate on the bottom. You can see that the pressure gradient that is needed to drive that flux through an N95 or a surgical mask is much higher. That means they're more difficult to breathe than the masks that we are designing. We do see some difference between the physical experiments and the CFD numbers in our plots. The reason being, in the experiments, we are using 3D printed filters. And with 3D printing, what happens most of the times is, out of those 300 pathways, some of them are occluded and blocked. So to drive the same flow, we have to impose a higher pressure gradient than what would happen in the CFD simulations, which is the idealized case. We see a similar trend in terms of the bulk pressure loss coefficient. Of course, the surgical masks and the N95s, they have a higher pressure loss. So does the HEPA filter that we tried. These are some designs that we have looked at. In general, you can see that as the design of the single pathway becomes more complex, the aerodynamics becomes more complex, and that helps in screening the particles out from the air. I'm going to just show some representative data for this particular design. It is Tortuosity 1.9. This is the pressure plot and the velocity plot for some streamlines. The more interesting thing is looking at the particle trapping trend. This is 3.5 micron particle. This helps us to figure out where the particles are getting trapped, which actually can help in improving the mask in terms of whether we can utilize the design to inactivate the pathogens. So this is the data on the filtering efficiencies between and a comparison between the CFD and the experiments. They're very similar. One might ask that here on the x-axis, we are just looking at 9 microns and bigger. What about the smaller ones? The thing is, we have had some 2 publications lately. We see that most of the viral load that is deposited at the initial dominant infection site, which is that red region in the nasal cavity model, the nasopharynx, that viral load is primarily carried by particles or droplets that are bigger than 9 microns. So we are trying to design a smart mask that can actually do a targeted screening of the bad droplets and not all droplets and aerosols. And we are trying to inactivate the pathogens that we are trapping as well. We have seen from the work from other groups that high temperature can inactivate the viral activity. So we are applying the high temperature boundary condition on the faces that trap more particles. The simulations are ongoing right now. We don't have much data, but we see that in terms of the pressure gradient or the pressure field in this geometry, there is not much difference with elevating the temperature to around 50 degrees Celsius on the high trapping faces. So that's it. We are continuing with the work, but in general, I think we think that this design of bioinspired filters could be applicable to other areas as well. For example, we can, 100 years back, the big challenge in front of us was water sanitation. I think over the next 20, 30 years, a big challenge would be air sanitation. And we can use these new ideas to develop filters that can sanitize the air around us. It can be the air inside a confined space like a car